MRI is a technique that's sensitive to water, and because we're predominantly made of water, it's great for imaging soft tissue inside the body. Especially the brain is largely made up of water. So because of this, we can get very nice images of the brain by putting someone in an MRI scanner.
MRI is used by using a huge magnet and aligning all of the protons, which are found usually in water within your body, in a similar direction, and when they move alignment, they give off a little bit of a signal, and this signal can be captured and taken in and processed as an image that you're probably often quite familiar with.
When water is placed in a large magnet, these water molecules behave like little compass needles, and they begin to spin together in phase. And then we can use magnetic field gradients and radio frequency pulses to manipulate these little compass needles that then give out radio waves, which we can then measure and use to generate an image.
So if you imagine, it's taking a snapshot of what's inside you and putting lots of these images together to make a three-dimensional shot of our brain.
Currently in the clinic, MRI is widely used to diagnose Alzheimer's disease. This is because it very nicely shows the atrophy of the brain.
So Alzheimer's is the most common neurodegenerative disease. Patients present with the early stages of Alzheimer's disease with impairments in their cognitive ability, and they have word finding difficulties and have a distinct loss in memories. So patients often have losses in sporadic memory, so they're unable to remember, for example, what they had for breakfast, but longer term memories, for some reason, are still maintained in these patients.
But the disease develops silently, perhaps 20 years before symptoms become apparent, and the disease starts with the build-up of these sticky proteins called amyloid in the space around your brain cells, and then as the disease progresses, eventually certain parts of your brain begin to shrink, and that's when you start seeing memory defects and other cognitive impairments.
When we scan these patients in the clinic, we see specific neurodegeneration, so shrinkage of the brain within the cortex, which is the part of the brain involved in cortical processing and thought, and also specific shrinkage in an area of the brain called the hippocampus, which is involved in the formation of memory. So the great thing about MRI is that it gives us really high spatial resolution, which means we can see at really in-depth detail the tissue within the brain. So we can look at volumes, for example, so we can look at the size and shape of the hippocampus and the cortex, and we can follow that over time. The great thing about MRI as well is that you can tune it to look at a number of different things. So for example, you can look at arterial spin labeling, which is an MRI technique to look at how much blood is being perfused into the brain. So in Alzheimer's disease, in areas that are dying, we see a reduced amount of cerebral perfusion, which is an indicator of neurodegeneration within the disorder. So it's the combination of all of these multi-modality MR images that give us a broad overview of the picture of neurodegeneration in Alzheimer's disease.
But the problem is that by the time they do this imaging, the brain has already started to shrink, and that is what we detect using MRI. So what I'm looking at specifically is one aspect of the pathology known as tau pathology. Tau is a protein that accumulates in the Alzheimer's brain, and I want to look at how the fluid that surrounds the brain, the cerebrospinal fluid or CSF, is responsible for the pathology. So I'm specifically using MRI to look at the movement of fluid around the brain and to detect these proteins in that fluid and to see if it's contributing to the accumulation of these proteins in the brain.
I want to develop the first non-invasive technique that uses MRI to measure glymphatic clearance. And this is a relatively new pathway that's being discovered that removes waste products from the brain, and it does this via the exchange of cerebral spinal fluid with the brain's interstitial fluid. And this acts to kind of flush out waste products from the tissue. I'm going to try and develop techniques that use MRI to measure glymphatic clearance and measure rates of exchange of cerebral spinal fluid with the brain's interstitial fluid.
So I'm working on an animal model of Alzheimer's disease. I'm using MRI to look at the animal model to see how the brain changes over time within this disease. So the great thing about MRI is that it gives you really high soft tissue contrast. And also one of the main things in terms of Alzheimer's research is that we can longitudinally scan the animals. So that means that we can image a cohort of animals at a really young age, image them again a month later, a month later, a month later, and leading that up. So then we can follow the pattern of atrophy, or the amount that the brain is shrinking over time, and see how that changes in these different animals. We can then intervene. For example, we can give the mice a treatment, for example, that we think is going to reduce the amount of hippocampal degeneration, and see whether we can follow that using MRI.
Those clinical images that you see are taking snapshots of the structure of your brain, but what I'm interested in is using new techniques to be able to see things that are moving within the brain. So this could be the blood flow, or also other fluids. If these fluids can be imaged using the MRI technique, if there's a breakdown or if there's a change in the movement of these fluids, it could be an indicator for particular diseases.
So I'm mainly working with cancer, and specifically colorectal cancer. And what I'm looking at is the vasculature of cancer, so basically how a cancer forms and then how it grows and create metastases that are cancer bits around the body. So I'm using different imaging modalities, but they are all within the main group of optical imaging techniques. The idea of this technique is to use visible light to look inside a sample, which can be a little bit tricky because we know that light inside the body is basically scattered and absorbed, so light cannot really travel through a body. So what I need to do in my experiment is actually render the samples transparent so the light can travel through them and it can be detected, so that we can form images of the internal structure of an organ or or a tumor. So optical imaging can be used both in preclinical research, but it can also be used in clinics in different ways. For example, we could take a biopsy of a tumor or just an organ from a person and then study that organ in really, really high resolution. So let's say we want to look at a specific tumor model like the colorectal cancer, then we could look at the vasculature inside that tumor with really high resolution. That would not be possible when the sample is still in vivo, so we couldn't look at the sample with these high details when the tumor is still within the person. If we wanted to look at a specific patient and a specific kind of cancer, then ideally we could take a biopsy of this person, then study the response of the tumor to different drugs and aim at administering this specific drug to that specific person. So this sample here is a colorectal cancer, and what I've done with it is optically clear it, so render the sample transparent so that light can pass through it. After this process of optical clearing, the sample is inserted in the machine, so this is an optical projection tomograph, and the sample is attached to the mount, and the mount will be immersed in this solution, that is the same solution that was used for clearing the sample. Then this is a motor that will rotate the sample inside the solution, and per each rotation, so per each angle, a projection will be acquired so that they can all be reconstructed and form a 3D image. So in this video we can see a colorectal cancer that has metastasizes in the liver. So all the blue bits are the healthy part of the liver lobe, while the yellow parts are the metastasis, so it is the tumor. And we can see how this system can show really, really high details of organs that developed cancer, so we can see single cells of tumor within the organ. This image was acquired by autofluorescence, so no contrast agents were used for looking at this kind of tumor. Basically, we just used a natural emission of light from the sample. And what we discover is that every kind of tissue emits light at a different wavelength, and in this case, tumor tissue emits light at a higher wavelength compared to the normal liver. And this is how, using different channels, we could detect the cancer within the liver.
So I'm mainly working with PET imaging, or positron emission tomography, and this works by detecting the gamma rays that are produced by the decay of a positron that's emitted by a specific isotope, and I'm mainly using an isotope called zirconium-89. So by putting a small amount of this into the cells become like a carrier for the radiation, and we can detect their position in the body by the emission of the positron and subsequently the gamma rays that are detected by the PET scanner. And this allows us to produce a 3D image of where the cells have gone in the body. So by tracking the cells where they go after transplantation, and perhaps if they migrate, we can find out if the cells have been transplanted successfully, and this can provide useful information in improving cell therapies. So this image here shows the distribution of cells in 3 dimensions in the mouse body. So it's overlaid with the CT scan, which provides the anatomical data. So we can see the bones and some of the soft tissue contrast. So in this case, some stem cells have been injected intravenously, and we can see that they've mainly gone to the lungs, which in this case is where we were trying to deliver the cells. In this case, the cells we're using have been modified to express an epitope which is used for the therapy of cancer, and hopefully these stem cells will be able to be used in the future to treat lung cancer. But to do this, they need to be delivered to the lung, and if this treatment was to be used in patients, we would want to know that they were getting to the right location, and we could obtain this information by imaging with a PET scanner. Because it's such a sensitive imaging modality, we can use very small amounts of radiation, and one of the things I've been investigating is the tolerance of the cells to the radioactivity. So I've been loading cells with varying amounts of radioactivity and finding the amount that can produce a good image, but at the same time doesn't produce any toxic effects in the cell. So there's sort of 2 aspects of toxicity which are important here. We don't want to kill the cells that are being used for the therapy, and you also don't want to damage the patient. Because it's a sensitive imaging modality, we can get away with using a smaller amount of radiation with PET imaging than we can, for example, with other nuclear imaging modalities that are based on imaging radiation.